Hello, I welcome you to part 3 of Neoplasia. I am Dr. Achal Danan and now we will be continuing. So, to understand the proto-oncogenes that are the cell cycle inhibitors and the checkpoint inhibitors, now we will understand the cell cycle. So, normally there is a M phase, that is a mitotic phase of the previous cycle that will lead to development of one cell, and this cell, the left cell, will lead to the G1 phase will lead to centrosome duplication. Then they will lead to G1S phase. This is a checkpoint, that is G1S checkpoint. What it will check? It will check the normal DNA content, that will it is having any damage or there is anything need to stop the cell cycle. It is to be evaluated by G1S. So what happens if it is damaged? So when it is damaged, the DNA repair gene will tell P53. So what will happen? What will P53 do? P53 has a regulation on P21. So with the help of P21, it will inhibit the cell cycle. So to understand this, we need to see that there are cyclins D that binds with cyclin dependent kinase 4 and 6. Then there is cyclin A that binds with 2 and 1. Then there is cyclin dependent kinase 1 that binds with cyclin B. This 1 and B is needed for G to M transition, and cyclin D with CDK4 is very important as it is required for G1S. The G1S is the most important checkpoint of the cell cycle, and mutation at this point or damage at repair at this point leads to malignancy. Although it can leads to malignancy at other checkpoints also, but this is at high risk. So there are two families of inhibitor. The first family is P16, P15, P18, P19, that is very specific to cyclin D with CDK4. Especially it is very specific to cyclin D. This one and this two, both it is very specific. Whereas the other, that is P21, 27, 57 are non-specific. It will be inhibiting all the cyclins and cyclin dependent kinases, and this non-specific P21 are in regulation with P53, that is stimulated whenever there is any damage to the cell cycle or to the DNA material. So now we will see if it is past the repair mechanism, how it gets further into the cycle. So there is one thing known as retinoblastoma. This retinoblastoma is normally bound to E2F. When the cyclin CDK4 with cyclin D regulates its phosphorylation, and retinoblastoma is phosphorylated, it leaves behind E2F, that leads to progression of the cycle from G1 to S phase. And this is very important, and if this is the mechanism by which the retinoblastoma actually keeps in control of the complete cell cycle and do not leads it to keeps it in control and do not let it in for continuous division. So if there is some mutation that is increasing the function of all these cyclins, that will lead to malignancy, whereas there is any mutation that is reducing the function of these inhibitors will lead to malignancy. So the bad is increase in function of cyclin D and cyclin D CDK4 and reduce function of inhibitors. So now we come to the topic where we were discussing